Now welcome back children. As I told you, till my previous class I have given you a background about what is DNA fingerprinting, what is the basis of DNA fingerprinting, what are VNTRs, and what are restriction enzymes. Now it is time to know about the stages or the steps of DNA fingerprinting. So if you are ready, here we go. So let us go step by step. What is the first step? First step is I want the DNA of the individual. So the source of DNA can be blood, it can be hair, it can be saliva, semen, body tissue, and from there we will be collecting the samples of the DNA. WBC, blood, semen, saliva, vaginal swabs, etc. They can be the source. The amount of DNA must be minimum 1 microgram. Minimum 1 microgram DNA मिलना चाहिए. तभी it is easy to study those VNTRs, right? So the first step in the process of DNA fingerprinting is to get DNA from any source. Once you get it, you need to isolate that DNA from the cell. Actually, first you need the cell of that individual, then the cell needs to be broken down, centrifuged so as to release the DNA. The DNA source is exposed to high-speed refrigerated, ultra centrifugation. This separates the nucleus, breaks the same to free the DNA. पहले तो हमने cell लिया, cell लेने के बाद cell को centrifuge किया so that the nucleus breaks and the DNA is set free. Now what we'll do? If you have a very small amount of DNA, आपको crime के side से you got a very little blood sample, बहुत कम DNA से काम नहीं चलता. In that case, what we do, we increase the amount of DNA by a process called as PCR. I mentioned PCR in my previous lecture and I told you I'm going to teach you in detail in biotechnology. तो सबसे पहले अगर DNA का सैंपल कम अमाउंट में हमें मिला है तो हम उसे PCR से उसका अमाउंट हम बढ़ाएंगे. Right? Once we have taken out the cell, taken out the DNA, made multiple copies of it, now and we have also separated it from the bulk DNA, and now we have those satellite regions. Now that DNA is going to be cut with the help of restriction enzyme. Say this is the VNTR I'm looking for, GATC, 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 GATC. You know that this repeat sequence will be found in the entire population, but the number may vary. In one individual, the number may be 4. In another individual, it may be 2. In another individual, it may be 3. Right? But you know that we take mini satellites which are actually guarded by restriction sites. So the restriction enzyme will be able to cut them, and इस तरह से आपके ये जो repeat sequence है इसके टुकड़े हो जाएंगे. Right? So the DNA is cut into fragments using restriction enzyme. Each restriction enzyme cuts DNA at a specific region. With the help of site recognition, the DNA is cut into separate VNTRs. So देखिए सारे VNTR आपके separate हो गए. ठीक है? Even आपके body में these VNTRs may vary, from one chromosome to another. आपके chromosome 1 में maybe ये 5 times है, maybe 6th chromosome में ये 7 times है, whatever. Even in between the two DNA, I told you, the number of VNTRs vary. तो आप उसके पहले टुकड़े कर लेंगे. ठीक? Now what will you do is the section of DNA that is cut, जो छोटे-छोटे टुकड़े कट जाते हैं, these are called as restriction fragments. Now this will yield thousands of restriction fragment of different sizes. ठीक है? तो आपके हजारों ऐसे टुकड़े बन जाएंगे, कोई छोटा, कोई बड़ा. Now you need to actually separate those fragments. You need to separate the longer fragments from the shorter fragments. Now how will you do that? आप देख रहे हैं कुछ fragment बड़ा है, कुछ छोटा है. Now how will you do that? So we will separate those fragments with the help of a process called as Aggro Gel Electrophoresis. I had told you about this in the previous lecture, and I told you in biotechnology I'm going to teach you in detail. Now gel electrophoresis is a method whereby these DNA, they are kept in small wells, and this side DNA is negatively charged, so the other side we have the positive electrode or the anode. So the DNA tries to move towards the positive electrode, but it will filter out as per the size. The bigger DNA will be at the back, the smaller at the front, and so on. So this is how the DNA fragments are separated. Once these fragments are separated, how will we see these fragments? So we use a dye. The name of the dye is ethidium bromide. It's not written here. I'll be telling you in biotechnology. And एक डाई डालते हैं इसमें, स्टेन जिसको हम लोग बोलते हैं, ताकि जब हम स्टेन डालने के बाद इसमें यूवी रेडिएशन डालते हैं, अल्ट्रावायलेट रेडिएशन डालते हैं, देन दीज फ्रैगमेंट्स दे बिकम विजिबल। ठीक है, तो इस तरह से हमने क्या किया, वी टुक अ पर्टिकुलर क्रोमोसोम, देन वी सेंट्रीफ्यूज्ड इट, वी सेपरेटेड द बल्क डीएनए, वी सेपरेटेड द सैटेलाइट डीएनए। दिस सैटेलाइट डीएनए का VNTRs वी कट विद द हेल्प ऑफ restriction enzyme. Now these cut regions, they can be of different length. We separated them with the help of a process called as Aggro Gel Electrophoresis. Right? Now once they are separated, what will happen? DNA is negatively charged, so it will be attracted towards the positive electrode. The shorter fragments will come forward and the larger fragments will be on the back. ठीक, अब हम क्या करते हैं, हमको पता है DNA double stranded है, right? So हमारे DNA ऐसे separate हो गए, the larger ones are here and the shorter ones have come आगे, right? And the still shorter ones have come आगे. Now we need to separate both the strands of DNA. DNA तो डबल स्ट्रैंडेड है ना, क्रोमोसोम्स, वी हैव टू क्रोमोसोम्स, वन क्रोमोसोम एंड अनदर क्रोमोसोम, एक क्रोमोसोम का डीएनए दूसरा का, नाउ वी नीड टू सेपरेट देम, राइट? सो हाउ विल वी सेपरेट द टू स्ट्रैंड्स ऑफ द डीएनए, राइट? नॉट द टू क्रोमोसोम्स, बट वी नीड टू सेपरेट the two strands of DNA, not homologous chromosome. I'm not talking about homologous chromosome, DNA double stranded होता है, right? So they are separated with the help of a chemical called as alkaline phosphatase. ठीक है, उससे आपके दोनों स्ट्रैंड्स अलग हो जाते हैं। Now once they are separated, I need to find out कि जैसे मुझ में हमने क्या किया था, मेरा सपोज क्रोमोसोम 1 लिया हमने, then what we did, we centrifuged it, then we separated the bulk of the DNA, we took out the mini satellite, the satellite DNA। अब मेरे बॉडी में भी this VNTR अगर क्रोमोसोम 1 में ही देखे तो हो सकता है एक बार 100 टाइम हो, फिर थोड़ा रुककर मे बी 50 टाइम्स हो, फिर थोड़ा रुककर 25 टाइम्स हो। सो द सेम VNTR विल बी ऑफ वेरियस लेंथ। ठीक है, अब ये अलग अलग टुकड़ों को अलग करना है, अलग करने के लिए आई पुट इट ऑन Aggro Gel Electrophoresis and these they got separated. Now whenever you take out the same chromosome from my body, that is chromosome 1, and then you undergo this same process, the same length of DNA will be formed and the same way they will be separated. This will be a fixed pattern, एकदम fixed pattern है। फिर मेरी बात समझो, क्या हुआ, आपने मेरा क्रोमोसोम 1 लिया, क्रोमोसोम 1 को लेने के बाद उसके डीएनए को सेपरेट किया, द बल्क डीएनए जिस पे काम नहीं करना अलग किया, जो जिस पे काम करना है दे सेपरेट एज थिन बैंड्स, दीज आर कॉल्ड एज सैटेलाइट, इनको लिया। नाउ इस पे एक पर्टिकुलर VNTR पढ़ना है मुझे। Now this VNTR in my first chromosome, it is found in first chromosome many times, many places, many times. हो सकता है शुरू में it is 50 times repeated, गैप के बाद maybe 25 times, गैप के बाद maybe 10 times. तो जब हम उसको restriction enzyme से काटेंगे तो मेरे फर्स्ट क्रोमोसोम से कैसा निकलेगा, you will get 125 लेंथ का, then you will get 50 लेंथ का, 125 लेंथ का, 110 लेंथ का, and electrophoresis से जब अलग किया तो they are separated. Now every time when you study my first chromosome, right, every time when you study my first chromosome, they will be separate and you go through the same process. The VNTR will be separated in a similar manner, 25 50 25 10 50 25 10, चाहे तो मेरे semen से ले लो, it can be taken from blood, it can be taken from any part of the body, from the skin, maybe saliva, if it's a male it can be from semen, anyways अगर आप कहीं से भी लेते हो, अगर आप क्रोमोसोम 1 को स्टडी करते हो और ये प्रोसेस को दोहराते हो, दैट मीन्स you centrifuge it, you remove the bulk of the DNA, then the satellite DNA you cut it with the same restriction enzyme, so it will form the same length, it will form a length of 50, a length of 25 and a length of 10. That means this pattern will remain same. But if you take it from another individual, from the first chromosome, maybe the first length that appeared maybe of 70, the second maybe of 20 and the third maybe of 5. So obviously this pattern will not be same. So how to see this pattern कि ये कैसे कैसे आया है, इसके लिए we use a technique called as sudden blotting. तो sudden blotting में हम क्या किए, पहले तो electrophoresis से हमने DNA को अलग किया, then DNA is double stranded, alkaline phosphatase से हमने separate किया. So अगर मेरा वाला देखे तो there is one of 50, there is one of 25, one is 10, DNA is double stranded, alkaline phosphatase यूज किया तो अलग हो गया, 50 50 का अलग, 25 25 का अलग, 10 10 का अलग। अब मुझे पता कैसे चलेगा, so what we do is sudden blotting. Sudden blotting में हम क्या करते हैं, हम एक नाइट्रोसेलुलोज मेम्ब्रेन लेते हैं। नाउ आपका जो इलेक्ट्रोफोरेसिस में ये जो डीएनए सेपरेट हुआ, ये नाइट्रोसेलुलोज मेम्ब्रेन को उसके ऊपर रख देते हैं। सो इट जस्ट एक्ट्स लाइक अ ब्लॉटिंग पेपर एंड द डीएनए का कुछ सैंपल विल ब्लॉट एंड कम ओवर हियर, है ना, ये ब्लॉट करके एग्जैक्टली उसी जगह पर आ जाएगा। आई टोल्ड यू ना, दिस इज माय जेल इलेक्ट्रोफोरेसिस व्हिच आई शोड यू इन माय प्रीवियस स्लाइड। ठीक है, इसमें ऐसे डीएनए सेपरेट हुआ, आई विल टेक अ मेम्ब्रेन कॉल्ड एज नाइट्रोसेलुलोज मेम्ब्रेन एंड द मोमेंट आई पुट द मेम्ब्रेन ओवर इट, ये जो डीएनए जैसे जैसे सेपरेट हुआ है सेम जगह पर सम ऑफ द डीएनए विल ब्लॉट इनटू दिस मेम्ब्रेन एंड दे विल बी ट्रांसफर्ड टू दिस मेम्ब्रेन, राइट। नाउ once they are transferred, this technique is called as southern blotting, right? See this, in southern blotting, these separated VNTRs which are now single stranded, they are transferred to a nitrocellulose membrane or nylon membrane which is actually placed on the gel. देखो gel के ऊपर रखें so the DNA gets transferred. The procedure is called as southern blotting after the name of E.M. Southern. E.M. Southern ने इसको खोजा था, so we call it as southern blotting. ठीक है, अब हो गया। अब मुझे केवल क्या पता करना है, I just want to know that how many times it is repeated. मैंने कहा ना, say मेरे 3 fragments बने, one may be of 50 repeats, one may be of 25 repeats, say one may be of 10 repeats. How will I know that? So if I am studying repeat sequence, say ATC, and this is repeated 50 times. Here it is ATC, it is repeated 25 times. Here it is repeated ATC, it is say repeated 10 times. So how will I know that? अब हमने क्या किया, I am going to take radioactive probes. Now what are radioactive probes? अगर यहाँ ATC है तो इसका complementary nucleotide क्या होगा? It will be TAG. It is going to be TAG. हम यहाँ पर छोटे छोटे probes बनाएंगे TAG के, सिर्फ TAG के। I am going to make them and there in adenine, thymine and guanine I am going to use radioactive molecules. ठीक, अब हम क्या किया, ये मेरा नाइट्रोसेलुलोज मेम्ब्रेन था जिसमें दे हैव एक्चुअली बीन ब्लॉटेड 50, 25 एंड दिस। अब हम लोग क्या करेंगे, इस मेम्ब्रेन को एक टब, एक बाथ बोलते हैं, उस बाथ में ये रेडियो एक्टिव प्रोब TAG भरा हुआ है, उसमें रख देंगे। तो जहां पर भी ATC होगा वहां पर TAG जाकर क्या होगा, बाइंड कर जाएगा, इट विल बाइंड, right? And and it is radioactive, so the moment we pass UV radiation through it, what will happen? It will form distinct bands. तो इसका बैंड इतना बड़ा बैंड है, इसका बैंड इतना बड़ा बैंड है, इतना बड़ा बैंड है। तो जस्ट जैसे आपका एक्सरे प्लेट बनता है ना, आपका एक यूनिक एक्सरे प्लेटिंग बन जाएगा ऑफ योर VNTR का सेपरेशन। जस्ट सी इट वंस अगेन, ठीक है? आई एम टेकिंग लिटिल बिट मोर प्लेस so that you understand. So just try to understand this. See, I have a length of 25, 50, then I have a length of 25, and then I have a length of 10. I have put radioactive probes. Now these radioactive molecules in presence of UV radiation, they will shine. And they will shine and they will form a particular banding pattern. तो मेरे nitrocellulose membrane में, how will this banding pattern look? It will be like this for the 50, then it will be थोड़ी देर एक बार 25 का like this and 10 का like this. It is just like an X-ray plate. ठीक है, ये मेरा बन गया. You got it? Now what I am going to do is I am going to take off another person. एक और आदमी का लेंगे. Now अगर उस एक और आदमी में I need little bit more space, so let me take the entire wood for this reason. So I told you in me what happened. The repeat was 50. Then the repeat was maybe 25 times and then the repeat was 10 times. ठीक, आप में भी ये VNTR है in the first chromosome, but in you the repeat here is maybe 100 times, then maybe it is 60 times and then maybe 2 times, right? So obviously अगर हम अपना electrophoresis करेंगे, so how will it happen? Let me do electrophoresis of both of them. So this is 100 सबसे बड़ा है, सबसे पीछे रहेगा. Then this is the place where 75 will land. अच्छा, let us make this 75. It will be good समझने के लिए. Let us make it 75, 25, maybe 10 and let us make it 5. ऐसा लिखते हैं. आपको तो पता है ये ऐसे सेपरेट करेगा. So पहले 100, फिर 75, then 50, जिसमें 50 है क्योंकि जितना छोटा होगा वो आगे जाएगा. Then maybe उसके बाद जो 10 है and जो आपका 5 है. राइट? तो मेरे में बैंडिंग पैटर्न कहाँ बनेगा? Here I use radioactive molecules, so it will shine over here 50. Then it will be, sorry, I should have taken here 25 also. So in me, my second band will be formed here because I have 25, and my third will be formed here because I had 10. In you, right, yours first one was 100. So आपका तो 100 वाला तो बड़ा है ना, तो यहाँ पाया जाएगा, पीछे पाया जाएगा. Then you had 75. 75 उसके बाद का ये इधर पाया जाएगा. And third you had 5, तो यहाँ पाया जाएगा. राइट? तो ये आपका एक यूनिक एक्सरे प्लेट बन गया, सॉर्ट ऑफ मेरा एक अपना प्लेटिंग बन गया. नाउ आई जस्ट वांट टू सी कि मेरे बैंड्स कहाँ बने एंड आपके कहाँ बने. हाउ विल आई नो इट? Right? So I will do it is, I will actually tag it with radioactive tags. If my, original sequence, the repeat sequence is ATG, as I told you, I will take radioactive TAC. So जहाँ ATG है, the TAC will bind. Then I will pass UV radiation. Once I pass UV radiation, they will appear like bands. तो मेरा बैंड कहाँ बना है? मेरा बैंड बना है in the region of 50, 25 and 10. आपका बैंड कहाँ बना है? In the region of 100, 75 and 5. So it is simple like X-ray plate. अगर आपका प्लेट क्राइम साइट में जो पाया गया उससे एग्जैक्ट मैच कर जाता है, that means you are the culprit. अगर आपका ये प्लेट नहीं मैच करता, that means you are not the culprit. So this is how we do it. Now I will make you understand the process once again. इससे simple चीज करना क्या है? I basically want to tally the DNA of two people. I'm not going for the entire DNA. I'm not going for all the chromosomes. I'm just going for a specific chromosome. उसमें भी I'm going only for the introns, and introns maybe I'm going for a particular VNTR. I know that in chromosome 1, this VNTR will be present in the entire population, but the length and number of VNTR across that chromosome will vary from person to person. Right? So हमने क्या किया? ये क्राइम के साइट से हमें मिला जो सेल, उसको हमने निकाला. Then first chromosome को we remove the DNA, bulk DNA, then the satellite DNA. Now this DNA में I just want to see ये VNTR कितना है और क्या लेंथ का है. So what I did was I used restriction enzyme, and then in order to separate this DNA, I used Aggro Gel Electrophoresis. Aggro Gel Electrophoresis में क्या होता है? The DNA separates according to its length, right? तो जितना बड़ा होगा it will be at the back, and जितना smaller होगा वो आगे होगा. Now how do I understand ये कितने length का है? So actually what happens? This is called as sudden blotting. So what I do is I put a nitrocellulose membrane, and in that membrane I know that this is the range of 100, this is the range of 75, 50, 25, 10, 5, and then I put it like this. This will blot and come there. So on that nitrocellulose membrane, I know that if the DNA is placed here, that means it is 50 repeat. That is graded. ठीक है? अब ये कहाँ पर हुआ nitrocellulose membrane में कैसे पता लगेगा? I will take a tub. There I will put what? The radioactive probes. So if it is ATG, I will take TAC radioactive. I will put it there. So obviously जहाँ पर भी ATG होगा radioactive, this will bind. Then I will pass UV radiation, so they will form bands. By looking at the band, I will come to know how many base pair long के आपके VNTR बने थे, and एक particular plate develop हो जाता है. This is from the crime side. अब जिस पे शक है, सबका क्रोमोसोम 1 लेंगे, we'll go through the same process. सबके सेपरेशन के बाद एक यूनिक प्लेट बनेगा. If this plate matches exactly जिस आदमी का this pattern of separation matches with that of the crime side, we will identify that yes, he is the criminal. So we have come to know actually how it happens. So let's just look at this once again. The separated VNTR, that is the single stranded sequences, they are transferred to nitrocellulose membrane. This process is called as sudden blotting. After this, what we do is we make radioactive tags. These radioactive tags are called as probes, DNA probes. ये question आया इस साल medical में. Now the nylon sheet is placed over this nylon sheet is immersed in a bath. In this bath, those DNA probes are attached. These probes will get attached to the VNTRs. Having the complementary nucleotide sequence. Then we will pass it through UV radiation, and then a particular banding pattern will actually appear, Right? Now we are going to match the banding pattern. In this pattern you can see the bands contain shorter DNA fragments and the bands will contain the longer. So the shorter and longer, यहाँ से यहाँ अगर देखोगे, this is the starting point and the ending point. So the larger are here and the smaller are here. So a very quick look. First you took the DNA, you cut it into pieces. Then you separated it from the bulk DNA, that is a satellite DNA. Now you cut it with restriction enzymes. Then you passed it to the gel electrophoresis. Now these double-stranded were then made single-stranded with alkaline phosphatase. Then the nitrocellulose membrane was placed over it. Then the radioactive probe was placed over it. And then UV radiations was put so that you could actually see the patterns. So there is another diagram explaining the same thing. So now we will look into the diagram from your NCERT. Now this is a wonderful diagram, after which I am sure your concept will be thoroughly cleared. See, it is showing that there is this is a DNA from individual A and this is DNA from individual B and this is the DNA from crime site. ठीक है, ये crime site का DNA. Now in crime site का DNA we are actually studying chromosome 7, chromosome 2 and chromosome 16. ठीक है, ये जो क्राइम साइट से हमको मिला. In chromosome 7, the repeat sequence that we are studying, in one of the DNA it is 11 and the other it is 7, right? So obviously if you look at here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, तो ये 11 वाला 11 के एरिया में बैंडिंग पैटर्न बनाएगा 11 में and 7 वाला बनाएगा in the 7, right? Now the same repeat sequence was also found in chromosome 2. In chromosome 2, in one of the DNA, you know that I'm getting one chromosome from mother, one from father. So in one it was 1, 2, 3, 4, 5, 6, 7, 8. So obviously the banding pattern will be formed at the 8 region. And the other one it was 7. No, sorry, 8 nahi. It is 1, 2, 3, 4, 5, 6, 7. So obviously it will be found in the 7 region. Okay, so one was in the 7, 1, 2, 3, 8 and one in the 7 region. Now let us come to the same VNTR was found in chromosome 6. 8 में था, it is 1, 2, 3, 4, 5 times repeated, 1 में 2. So here ये देखो ये 5 के बगल में pattern बनाया और ये 2 के pattern में बनाया. So a particular type of pattern has been made. Now these are two individuals जिस पे हमको शक है, right? In the first of all, in this person, in chromosome number 7, in chromosome 7, it is 3 and 4 times repeated. ठीक? So this is person 1, person A. तो यहाँ पर किस में red has come in 3 and 4, 3 and 4 में. Look at this 3 and 4 में. Now इसमें क्या है? It is 3, 6 to 8. So it is just इसके जैसा 8 and 7. ठीक है? Just like this. And इसमें क्या है? It is 5. So this is found in the region of 5. And in the other it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it is found in the region of 10. ये इसका pattern बना. Now this is the other person, its pattern and its pattern is exactly the same. So the DNA has been separated, the VNTRs have been separated at the same place. So now by comparing these X-ray patterns, we can clearly say that this person is the criminal. Right? So there is a wonderful diagram in your NCERT as well. So I hope with this, the process of DNA fingerprinting has been well explained and you have understood. We will just go to the two significance or application of DNA fingerprinting. One, you know that DNA fingerprinting can be used for forensic science in maternal-paternal disputes. Now, like skin fingerprinting, DNA fingerprinting can distinguish one from another. While skin fingerprint can be altered, there is no known method to alter DNA fingerprint. It remains the same in every cell and every organ of a person. Every child will receive 50% from the mother and 50% from the father. Thus, by comparing the VNTR sequence, this is the child VNTR sequence. How will I identify the mother and the father? So one of them will be actually identical to the mother. This is the father and one. So you know, इसके बच्चे के पास जो है, some of the pattern will be similar to the mother and some of the patterns will be similar to the father. इससे हम लोग पता कर लेते हैं because one we receive from the mother and one we receive from the father. And the second application is, of course, in solving crime sites. I showed you with the NCERT example how it is helped in solving crime sites. So that's it. Now you know NCERT मैंने almost पढ़ाते वक्त PPT में ही डिस्कस कर लिया है. Many of the slides were just actually, paragraphs from the NCERT, but still, as a protocol, I would like to visit NCERT once and show you कि क्या चीज कहाँ है. So over to NCERT. Please open page 121 of your NCERT. There is the topic DNA fingerprinting. Now look, we'll go quickly, very quickly, paragraph wise. In the first paragraph, you can see it is just saying that 99.9% of the base sequences are same in humans. So there is only a difference of 3 into 10 to the power 9 bases. Now every time if you want to compare 3 into 10 to the power 9 bases, this will be a daunting and very expensive process, right? So that would not be possible. So what we decided was that we will go for a segment of DNA only. Now that segment of DNA which is compared or used in DNA fingerprinting, that's called as repetitive DNA. Now this repetitive DNA is separated from the bulk genomic DNA during density gradient centrifugation. So it is called as satellite DNA, which can be AT rich or GC rich, and the length and the number of repeated units. The satellite DNA is again of many type like microsatellite, mini satellite, etc. I gave you a detailed account of all these types. These sequences normally they do not code for any protein. Why? Because they are a part of introns, but they form a large portion of human genome. They show high degree of polymorphism and thus they form the basis of DNA fingerprinting. Right? Now since DNA from every tissue like hair, blood, skin, bone, saliva, sperm of an individual show the same degree of polymorphism, they become a very useful tool in forensic applications. Right? Further, since they are inheritable from parents to children, they are also the basis of paternity testing in case of disputes. Right? So this is now in the next paragraph, it speaks about polymorphism. Now you know that polymorphism is the basis of genetic engineering. Now what is polymorphism? It is trying to say. So how is polymorphism actually created? So it says that any new gene arises due to mutation. Now this mutation can be in somatic cell or it can be germ cell. If the mutation is in the germ cell, does not impair individual's ability to have offsprings, and it can transmit the mutation and spread to other members of the population. Now when I talk about spread to other members of the population, in what amount? I told you in frequency greater than 0.01, then we said that a new allele has come and that allele is polymorphic. Right? Now since this इसमें ये लिखा है polymorphic DNA, why it is found more in introns. So this you can find it in this paragraph. So one of the DNA that we use, that satellite DNA which we actually use in DNA fingerprinting, one of them, the one that was used by him, that is Alex Jeffrey, that was VNTR, that is called as variable number of tandem repeat. Right? So he used a satellite DNA as probe that shows high degree of polymorphism, that he named it as VNTR, variable number of tandem repeat. Now what were the steps of DNA fingerprinting as taken by him? It included the following steps. You remember in my discussion, the steps were, in my discussion, I gave you the steps elaborately. But when it comes to the school exam, let me tell you, this is a very, very, very important question for your board exams. And board exam mein, agar aapko aayega, give the steps. Please do not write anything which I have taught. Please do not write it. Just write these 6 points. It will come for a 5-mark question. Even in your school it will come. First, isolation of DNA, then digestion of DNA by restriction enzyme, then separation of the fragment by electrophoresis, then transferring of the separated DNA to synthetic membrane nitrocellulose, जिसको हम लोग कहते हैं southern blotting. Right? The process is called as southern blotting. आपके NCERT में the word southern blotting is not given. Then hybridization using VNTR probes. You used small fragments of DNA which were radioactive. Then detection of the hybridized DNA. Now कहाँ पर ये radioactive DNA hybridize किया है, कहाँ पर लगा है, that you found out by autoradiography. Right? So these are the steps. Please, once again I insist, whenever it comes in your board exams, do not write what I have given. Just write these 6 points and you will get your perfect score. right? Now in the next paragraph it has described about VNTR. So what is VNTR? It is a class of satellite DNA which we call as mini satellite. Take here the nucleotides are tandemly repeated in many numbers. The number of repeats shows high degree of polymorphism. The size may be from 0.1 to 20 kilobases. They can be easily hybridized with VNTR probes. So this is what we do it. Now this will be every DNA is unique except in case of monozygotic or identical twins. Right? So this is about the VNTR. Please read through that, and then in the next paragraph or the last paragraph, it just speaks the application. It says besides the two applications which we have already spoken, that is paternity dispute, if at all it is there, and crime, then this DNA fingerprinting can also be used in other cases like determination, population and genetic diversities. So this is another third application which is given in your NCERT, and this is the diagram which I actually gave you as an example, right? So with that, the topic DNA fingerprinting has come to an end. A very quick recap: DNA in skin fingerprinting we can find, but by surgery it can be manipulated. But in case of DNA fingerprinting, there is no method. I need to compare DNA. I am not going to compare the entire DNA. I am going to compare only introns, maybe repeat sequence, and that too only of select chromosomes. Now these repeat sequence, they are found in the entire population, but the number of repeats and the frequency of occurrence, it varies from person to person, right? So I am going to take a particular chromosome, separate the other DNA and this satellite DNA, and I want to know mere mein that VNTR is how many repeats and how many times, right? So I will first cut it with restriction enzymes, separate it by gel electrophoresis. According to its weight or the size, it will get solved. Now how to know it? Alkaline phosphatase, say first I will make it single stranded, then I will put it on a nitrocellulose membrane, that is southern blotting, and then put the probes, the radioactive probes as a result, and then put UV radiation. So where the banding pattern has been made, that means it is in the number 75 or 25 or 10, that will be clearly visible. A complete plate is ready. अब जिस पे शक है उसका भी you go through the same process, compare the plates, and thus paternity disputes and crimes, they can be easily found out with this method. So I hope the topic is very clear. With this, the chapter also comes to an end. I hope you enjoyed. There's a video for every topic. Whatever you find a problem, please go through that. In my next lecture, I'm going to discuss 10 years board papers from this chapter, what type of questions they come, so that you can score the perfect 100 in your board exams. And in the next paper, I'm going to discuss 11 years medical paper from this chapter, so that you know that whatever you studied, is it sufficient for medical or not, and how to study. So with those words, I just sign off today. Again saying, please go through the chapter thoroughly, a very important chapter for board and medical, right? Keep studying. We'll meet in the next video with the question papers. Till then, good luck and good